So good morning, everybody. We begin today's unique webinar. The topic for today's webinar is bioinformatics approaches in genomics and proteomics. And we are very delighted to have with us the Honorable Vice Chancellor and our main Professor Indrani, who is the research director, who worked as a research director Nitte University, and our resource person Amit Kumar Yadav and Mr. Shyam Parabu, who are great innovative spirit in their own field, and we have a lot to learn from them, and ultimately it's going to benefit our university and our students, and we are very delighted that the panelists, the expert speakers are here with us. We begin today's workshop. So bioinformatics, as we know, the objective of this webinar is it is used very vastly in research field, and it would be very advantageous for all the students to gain the knowledge from bioinformatics. So for this purpose, we are going to conduct this unique webinar. We are very honored to have with us our Vice Chancellor of Nitte University, Professor Dr. Satish Kumar Bhandari with us. To introduce him, he is an accomplished ENT surgeon and has held various posts as Vice Dean and Dean of K.S. Hegde Medical Academy. To add a feather in his cap, he was awarded the fellowship Addendum Gradum from Royal College of Surgeons of England in the year 2018. He has contributed immensely to scientific advancement in his field of specialization. He has more than 73 publications and has attended more than 80 national and international conferences and has given more than 25 presentations at conferences. He has diverse interests which led him to publish 2 books, one in his own specialty and the other on collection on rare medicinal plants of coastal Karnataka. Professor Bhandari also holds a patent entitled Mouthwash for Managing Radiation-Induced Oral Mucositis in Patients with Head and Neck Cancer, which was published in the year 2020. He was conferred the Lifetime Achievement Award by the Association of Otolaryngologists of India, Karnataka, in the year 2019. He has a long list of executive and non-executive posts, such as President of IMA, South Canada branch, President of Association of Otolaryngologists of India, Karnataka chapter. Dr. Bhandari was also instrumental in establishing the Mangalore Laryngectomy Club as K.S. Hegde Medical Academy, Mangalore, which is a registered club. He is a recipient of various honors and awards, the most prestigious one being the Aryabhatta Award for Excellence in the field of medicine by the Aryabhatta Cultural Association, Bangalore. Distinguished Alumni Award from the Manipal Academy of Higher Education, Manipal, and the Eminent Education Award by the National and International Compendium, New Delhi. His other achievements include the Samaj Ratna Award in Medicine by the Jnana Mandira and Sanskriti Trust, Bangalore, Saurabha Ratna Award in Medicine by Suma Saurabha State Awards, Bangalore. Not many people what they know him is he's a keen proponent of Yakshagana, a popular traditional folk dance in coastal Karnataka. He was awarded the Achievement Award by the Mantra Natyakala Academy, Mangalore in the year 2018. To add to this, he was awarded the Swami Vivekananda Excellence Award on in the month of March 2019. We bring to you this multifaceted Honorable Vice Chancellor, Professor Satish Kumar Bhandari. Now I request Professor Bhandari to give the opening remarks.
Good morning. It's my privilege to be part of this interesting webinar on bioinformatics, approaches in genomics and proteomics. My esteemed colleagues, Professor Dr. Indrani Karnasagar, Dr. Rama Adiga, and my colleagues from the Nitte University, and the very learned resource persons, Dr. Amit Kumar Yadav and Dr. Shyam Parabu, and all the participants in this webinar. Thank you very much for participating in this webinar. Greetings from Nitte University. For the external resource persons, Nitte University is a very young university, 12-year-old university, It's mainly health related university with our flagship constant college is our medical college is with its 1,000-bed hospital with 21 rural health centers, catering to the needs of more than 5 lakh patients annually. And most of the treatment is given as a subsidized cost and also ably assisted by our other constant colleges like medical colleges assisted by dental, pharmacy, then communication, architecture, nursing schools, and physiotherapy school, and also our research facility, exclusive research facility in the form of NUCSER, what we call Nitte University Centre for Science and Education. These are the places where we have developed state-of-the-art facilities for particularly cancer research, neurological research, and communicable disease research, uh, and uh, and well equipped with the labs like which are dealing with the proteomics and genomics. Uh, Very apt that the NUCSER has taken up this particular subject for this webinar, bioinformatics, approaches in genomics and proteomics. As you all know, bioinformatics is an interdisciplinary field that develops methods and software tools for understanding the biological data. In particular, when the data is very large and very complex, I think bioinformatics is very much essential to coming, particularly in the field of genomics. This is very useful, and it is interdisciplinary, as the name suggests, combination of biology with the computer science, information engineering, mathematics, statistics. Is everything is collaborated here, and it's really the interdisciplinary subject. And it came to limelight, I think, with the whole genomic studies in the 1950s, 1990, I think, I believe. After that, it came to limelight. I think most of the people started using it. Unfortunately, I am not very happy that it's being well handled or used in our universities and our colleges. Otherwise, we would have had a lot of publication by using bioinformatics. But I am very happy that this particular topic by our senior scientist Indrani has addressed it by getting some people who are well equipped to handle this area. And there is lot of things can be done by using bioinformatics, particularly our data, which is available in the hospital. When we are dealing with the more than 1,200, 1,300 patient outpatient and more than 600, 700 inpatient, with varieties of cases and a rich source of medical knowledge should not go waste. I think by using bioinformatics with the help of our state-of-the-art laboratories, we have established 7 laboratories in the medical school and in the hospital and in the NUCSER. And recently we added Technology Enabling Center, where our Indrani Karunasagar is the director, and we are doing quite well for the last 4 or 5 years in the field of research. I think the bioinformatics is going to be a feather in the cap in the university if you develop it well. I congratulate Dr. Rama Adiga and her colleagues for selecting this topic and inviting the very renowned faculty, Dr. Amit Kumar Yadav, Senior Research Scientist, Translation Health Science and Technology Institute, and Dr. Shyam Perugu, Assistant Professor, Department of Biotechnology, National Institute of Technology, Warangal, for this webinar. I think their rich experience in this particular area will be beneficial to our students and faculty, and definitely our students will be benefited by this webinar. Very informative, going to be a very informative webinar, and I hope the participants will carry rich memories, enlightened by this workshop or webinar, and will be more enlightened or knowledgeable as for the bioinformatics. And also it will help the university in developing this particular area of bioinformatics and getting more publications, research by using bioinformatics in proteomics and genomics. which is very much essential in these areas to do research, and bioinformatics can give a lot of encouragement or fillip to this area. I hope all my colleagues will take it in a positive manner, in a right spirit, and use this webinar for enlightening or encourage to advancement of their knowledge in this particular area. Wish you all the best. May God bless you. Thank you, Rama.
Thank you very much for that wonderful message and the thoughtful message. It will certainly be taken, and we will put all our efforts and take forward the message and try to implement it in our university in a big way. Thank you very much for your support, and thank you very much for giving the wonderful message. Now I request Indrani Karunasagar, who is the director of projects NST, DST, New Tech, to talk about Professor Indrani Karunasagar. She's an internationally renowned scientist and also the director of UNESCO Centre for Medical and Marine Biotechnology. She holds a vice chair at the International Jury for UNESCO Life Science Prize and had worked as the Associate Director of Research in the Karnataka Veterinary and Animal and Fisheries Sciences University. Her career lasts for more than 35 years. She held a very important post as a Director of R&D, Nitte University, and also headed the Institute of Science Education and Research and was the Dean of Faculty of Biological Sciences for 6 years from 2014 to 2020. Her international achievements include, she was a visiting professor to the University of Warburg, Germany, University of Ghent, Belgium, through collaborative projects. She is the principal investigator of many national and international projects. She has a list of awards, and the list looks like a mini compendium. She was awarded the National Technology Development Award with the Government of India, the Sir M. Vishwesharaya Award for Excellence in Research and Lifetime Contribution, Rafiq Ahmad Kidwai Award given by ICAR, Lifetime Achievement Award given by the Department of Biotechnology, Punjab Rao Deshmukh Krishi Vidyapeeth, Outstanding Women Scientist Award given by ICR Government of India, the MS Swaminathan Award, and the National Bioscientist Award given by DBT Government of India. So we bring before you a very knowledgeable person with a vast knowledge, and she's going to give you the welcome address, and she's going to give you an insight into the present workshop. Thank you very much, ma'am.
Thank you, Dr. Rama, for that, I should say, elaborate introduction. First of all, I would like to welcome most heartily and express my deepest thanks to the Honorable Vice Chancellor, Professor Satish Kumar Bhandari, for whatever he has done in his capacity to raise the research, I should say, image of Nitte University. His interest in research and his, I should say, passion to take forward the university to an international standing is evident from the one word that he used, and that is interdisciplinary, which is actually the basis when we talk about the word bioinformatics. We're all aware of the subject of informatics being offered in engineering institutions. We are aware of biological sciences, medical sciences in the other universities. But what did this word bioinformatics do to bring about such a vast change in the concept of interdisciplinary? This is probably one of the first words that Earth. For me, it is, I should say, nostalgia as I think of this word. It was about 1990 when the Department of Biotechnology first set out to bring about amalgamation of these two sciences to set up the bioinformatics information centers in the entire country through the BTIS, and they supported setting this up through, of course, infrastructure and the training that they started to give. And I'm happy to state that by about 1996, we at the University of Agricultural Sciences then, where I was working as an associate professor, and Dr. Karunasagar, who was the professor, also could impress upon the Department of Biotechnology to give us a center, and the beginnings of bioinformatics were then with this center located at the College of Fisheries. As the coordinator of this center, well, I must say, year after year, on February 3rd and 4th, we met at various stations in India to discuss the progress that we are achieving. But there is also a sad story to it. While the government of India was promoting bioinformatics through setting up of these large number of centers, which resulted in many universities also starting the MSc Bioinformatics program, where people rushed to take admissions, which sounded as if India was on a very progressive path. We realized that this became short-lived, and in course of time, within about 10 years, there was a decline in the number of people seeking admission for bioinformatics, with many institutions in the country closing down their program because there were no admittees. Now, this state of affairs could have been avoided if probably there were predictive tools as it's available today in the form of, say, softwares or algorithms that have been developed. And COVID-19 times are the best to prove the power of bioinformatics. This pandemic has taught how India is in a way lagging behind, though we have the best brains in the country, of course, doing the best for the world, but we could have had a larger number of people specializing in it. So the shortage of bioinformaticians is responsible for the present state of affairs where we lag behind. While we had stalwarts like Professor Dharmalingam of Madurai Kamaraj University, Professor Alok Bhattacharya of JNU, who were the leaders in this field, helping the Department of Biotechnology to progress through the bioinformatics programs. We had Dr. Madan Mohan at Delhi DBT as the advisor. Having the best of bioinformatician. So having said this, how can we now look at this subject? Well, I have been asked to speak briefly about what is bioinformatics, though the advertisement says welcome address, but I was informed by the moderator to speak on the purpose of this particular meeting. So we need to know what is this. Now, the application of statistics and of course computer sciences to the field of molecular biology, where we had biologists and the IT industry coming together, has resulted in the study of, I should say, bioinformatics in its application of genomics and proteomics. Now, if you look at the subject of genomics, it does not make any meaning to have a series of alphabets placed one beside the other if we did not give a meaning to it. But the first step is to have the alphabets, and that is why genomics preceded the studies of the powerful proteomics that came about later. Now, in talking about genomics, a good example would be if you just wrote CAT, SAT, AND, RAT, all together, that would make no meaning at all if these alphabets were placed beside one another. But if you were to split it into the triplet codes and say cat, sat, and rat came or something like that, then you find that by splitting these alphabets, you are coming to a point of giving meaning to a series of alphabets placed one beside the other. So this precisely in layman's term would be what is genomics, and a functionality or making a meaning out of it becoming proteomics, functional genomics rather. Now, when this word was first coined in I think 1979 by Pauline Howick, and it remained always about, I should say, study of processes in biotic systems. But if we look till late 80s, the primary use was in the area only of genomics, proteomics, and then the omics era came with a lot of newer words, like we got to have transcriptomics, metabolomics, every omics that came about, epigenomics. Now, what this really means is there is a great scope, because through bioinformatics, there is, of course, the creation and advances that we are seeing in databases that are being created, and public databases have become the order of the day. Now, what better way to really speak of the advantage of public databases than during this pandemic, when the whole world has come together to solve problems related to the pandemic. And what are the problems? You need to, yes, have treatment, you need to have a vaccine, you want to trace the origin. Sometimes it's for political purposes, but that is not really the main thing. If you trace the origin and if you see how it will move forward, you will be able to also predict. So the word prediction is associated with bioinformatics. Now, when we talk about treatment, we have heard the government advertising for repurposing of drugs. You may have a drug for a particular condition, but how can it be used for this pandemic, this virus outbreak? Can be done through, say, drug docking studies. Now, the word drug docking itself would need to be analyzed by the use of several softwares. All of this has been made possible through the development of various softwares. Algorithms are written, the computational and statistical techniques are used for all of these purposes. So what was theoretically used to solve a problem has found great application during the pandemic. And maybe this was lacking when bioinformatics first got introduced. A lot of engineers were working on it, but people did not see the application of all of this to a larger good. And today, when we have seen the use of bioinformatics to find treatment, to develop vaccines, then we realize that this science is truly an inter and multidisciplinary science that has broken all the barriers between the different subjects. Now, when I say this, I'm sure many of the people in the field, of course, have read about vaccine development, which the whole world is now aiming at. But many of us may not know that within a week of the virus affecting and bringing about mortality, the whole virus was sequenced. And when the virus was sequenced, companies like, Moderna vaccine, which we are talking about, the vaccine was ready 1 month after the pandemic. Now, one could not have imagined that a vaccine would be ready within such a short period. Though we know about it only now, in the last 3-4 months, we've been hearing about it. But right during February, early this year, the vaccine already ready, the mRNA-based vaccine. Now, how was that possible? It was by the use of bioinformatics tools to design vaccine. Drugs, yes, as I already said, many drugs were being discovered, antiviral drugs, but you can't use all of the drugs unless you are able to show, can it dock in the regions, can it work on. Also, the mutations of this virus, how are they studied? They were done during the course of many sequences that were available. And we had our convocation sometime back, and I'm sure our Honorable Vice Chancellor will be able to endorse this view of mine, where the convocation address that was delivered by the Chief Scientist of World Health Organization, Dr. Soumya Swaminathan, one statement that was mentioned was, if the world had come together this way for many other important diseases like malaria, tuberculosis, or many other diseases that afflict African countries or other countries of the world, I'm sure we would have conquered them by now. And this is evident from the fact she mentioned that the vaccine that came about, the drugs that came about, were possible because of the use of various tools. In the former years, a vaccine took several years to be developed and lent itself to a lot of multi-country trials before it became available, and easily took a decade sometimes. But now it has moved so fast that since the outbreak, within a year, we're already ready with vaccines having undergone phase 1, phase 2, phase 3 trials. So the initial phase was very fast, that is the development phase. So we hear today about the Pfizer vaccine, we hear about the Oxford vaccine, the whole protein vaccines that by the Sinovac company, and also of course the Moderna vaccine. So how has all this become so rapidly available? It was because of these developments in mapping and analyzing the DNA available, aligning these and all of the protein sequences, and of course comparing them, creating, because we need to have something that would be globally acceptable, sometimes locally more relevant than at the global level. And for all of this, there are databases. In that connection, I wanted to mention also, of course, what the convocation address really delivered said. These databases have got all of these sequences, say for this pandemic virus, available and has been put into a database that could be extracted. So when in our own university, we started looking at certain mutations in the envelope glycoprotein gene of SARS virus. We picked it all out from the database and did bioinformatics analysis. But what prompted us is not just the theoretical part to do this study. When we found that the cases sometimes were missed out in some countries, like say in some states in India, like Gujarat, Odisha, as well as Madhya Pradesh and Tamil Nadu, they found that there were some mutations that were seen, and these were detected by looking at the sequences available in the public database. So we did not go and sequence all of the virus because the world has come together to deposit them, and such efforts have now become global. So you have databases for cancers, different cancers. You have databases for the different kinds of diseases where people are now putting all their sequences, which are available for everyone else to extract and use bioinformatics tools to develop diagnostics, to develop vaccines, to look at drugs for therapy. And the mutational studies will help us sometimes see how what works in one area may not work in another. And therefore, with two young speakers, I should say, Dr. Shyam and Dr. Amit, what we are really looking at is the future of this old field that has now become very young by experts who have realized the importance of the subject, and in combining the experimental tools with the theoretical tools that are there, the best results can be delivered. And what are some of these experimental tools? Yes, sequencing. Everyone cannot have facilities for sequencing, and therefore the government of India as well as internationally, there are sequencers available. For instance, in Beijing, there is a whole institute that is involved in only sequencing. And that is one reason why they are sometimes ahead in such ways. Then if you take countries like India, we have, yes, government institutions, but we have a lot of private players who have come into it. If you look at the volume of money that has gone into the business of bioinformatics, it's about $6,400 million. It was in 2015, but the prediction in the next 10 years until 2025 is at the rate of 13.5%, but it's going to be even more because of the pandemic that has broken out. So the business of bioinformatics has become very big, and these platforms have become inherent to many companies that are running the drug manufacture, that are running into vaccine manufacture, because the platforms will go from prediction, they will go then to experimentation, and then they will go to actually execution. And therefore, we are really at a very, very, I think, important phase of science today all over the world. And we need to remember, we are talking more and more about certain words like precision medicine. We are talking about individualized therapy. All of this will be possible. It is expected that by the next decade, like we carry our smart cards, each individual will have a chip having their entire genomic data, which will, of course, today the sequencing has been reduced to just about $4,000 per individual sequence, or even less. I'm stretching the volume and saying 4,000, but I heard there are some institutes, even in Pune, where it is done for $1,000. And this information will become very important for carrying, which will let a doctor decide which drug works for you, which drug doesn't work for you, what is your allergic status with a particular drug, or which is most effective. And towards this, I'm sure all of us have heard, many of us may not have also, about IBM Watson. IBM, as you know, is, yes, a computer company, yes, but IBM Watson is done for, say, cancer, especially the breast cancer. And one institute that was involved a lot in India, in Mangalore area, is Manipal, Kasturba Medical College. And IBM Watson enabled people to have which best drug would be suitable for an individual for breast cancers. But this had to be done through lots and lots of genome sequencing and having these personalized data in place. And therefore, you have a name, and sometimes a name gets repeated, as you know, very often if there is a shortage. So you have a large number of people with the same name, but the fingerprinting that we talked about earlier has now moved to genetic or genomic fingerprinting, where each individual is different from another. And the closest may be a pair of twins, but there also there is a lot of difference. And therefore, I think with some of these examples, I have meant to inform the youngsters who are online to listen to two powerful people on the application of a tool that, through the science of bioinformatics, is revolutionizing the world. I know we have techniques like, mass spectrometry. I will not talk much on it because the College of Pharmacy, the Pharmacy Institute of our university, conducted about a month ago a webinar on mass spectrometry, where I spoke about peptide sequencing and explaining the importance of mass spec in research, because there again I was only an inaugurator, so asked to talk on mass spec. But having been involved on the use of such techniques and having been recognized by the publications in these fields, because of which I tend to be called, I thought it is good for all of us to know that while you have advanced equipments to do the job, what we need to understand is the basic principles in each of them, and not to mechanically think. We have to become discoverers. And therefore, if there are equipments in place, if there are engineers who have built them, the biomedical engineers, the physicists, and a lot of other scientists who have got into this job of making softwares for us ready, of having databases which allow access to everybody, we need to realize we have come very close, and we've all realized that the more and more we know about any topic, or the more we think we know, we realize how ignorant we are, and we are a drop in the ocean of sciences, and have to play our role in letting everyone realize that we have learning will never end. We are students all our life, and bioinformatics is going to be the subject where one needs to put their heart and soul to see how just sitting in front of a computer, how powerful your research can be, so that it helps humanity in applying everything that we have talked about, whether it is the genome-wide association studies that we do, or modeling any for evolution, or tracing origin of a plant or an animal, or the virus. I talked only about virus only because I'm a microbiologist and I work with these small creatures. Understanding them is a lot easier than understanding the multicellular forms of life, and therefore I think I stand to be really humbled by the fact that there have been so many experts all over the world, and in India, names such as people from the Genome Integrative Biology Institute in Genomics Institute in Delhi, the CSIR Institute, the volume of work that they've been doing in genome sequencing to understand not just this virus, but a lot of other viruses and a lot of other human diseases, the genetic diseases, all of that needs a special mention. And I think Nitte University has made a beginning. And I'm sure all the young people who are there in this institute, I always think of the entire university as an institute, but what I really mean is in every institute of this university, the scope for doing is never ending. Sky is the limit, and even beyond sky, I should say. So thank you very much for this opportunity, and welcome again to Dr. Sham and Dr. Amit for having taken time off to come and speak to everyone on the tools that you've used to be where you are. Thank you, Dr. Rama, for organizing this work.
Thank you very much, Madam, for that important message, that in order to move forward, we need to learn from history and not make the same mistakes, and also to giving that important stressing the message of interdisciplinary nature of the subject. Next, we welcome our resource person, Dr. Amit Kumar Yadav, who is a senior research scientist at the Translational Health and Science Technology University, and he's a PhD in biotechnology with MSc in botany and BSc also in botany from Delhi University. His interests are in post-translational modification, and he has developed several tools for analysis of the PTM code, which we'll be listening to him shortly. His other interests are in big data in biomedicine and data visualization using computer science methods. He had several publications and various invited talks. He has also patents in the area of mass spectrometry. He has received several honors and awards, such as the DST Young Scientist Award and the GATE. He is also qualified in GATE. And the prominent award that he has received is the MIT Technology Reviewers Young Innovators Award, India, in 2013. He's also received the Innovative Young Biotechnologist Award in the year 2013. We welcome Dr. Amit Kumar Yadav for your webinar topic. Welcome, Dr. Amit Kumar. Yes.
Thank you, Dr. Rama, for having me here. Am I audible?
Yes, yes, you are.
I'm sharing my slides now. If they are visible, just let me know and then I can begin.
Yes, it's visible and you're audible too.
Thank you so much, Dr. Ramadiga, for having me here. And I congratulate Nitte University and a great message on the importance of bioinformatics by both Professor Bhandari as well as Professor Rona Sagar. And without much further ado, let us get started. And everybody talked about the importance of genomics and proteomics. And as also mentioned earlier, that proteomics stands on the sequences when we have from genome. So the topic I chose is educational one because unless people understand the basics of how mass spectrometry and proteomics works, it is tough to imagine how they can be used in their own research. So I wanted that people across masters, PhDs, and even faculties who are not well versed with proteomics should understand what are the basics of this technique and what it means to identify a protein in proteomics. So with that thought, I had chosen the topic to be proteomics and mass spectrometry from peptides to proteins. So this is a cartoon made famous right after the human genome was sequenced, wherein God is visibly stressed out that the hackers have cracked the genetic code. So what now? And he was being informed by an angel, as you can see. So this is something I tried to think about in detail during my PhD, and then I came up with the idea that no, this is not enough, and I made a small change to it. And where got it think perhaps not. They still have a dynamic proteome code to break, and they still cannot hit a moving target, which means the genome is static, but in different snapshots of time, the proteome may not be the same. The idea I'm trying to convey here is that even though an organism has one genome, but it could have many proteomes depending on the context. But sequencing proteins is not as easy as genomics. The advances in genomics are quite really much far ahead than in proteomics. And studying proteins using proteomics is quite an analytical challenge because of several reasons, of which the prominent ones are highlighted here. For one, there's a huge dynamic range of protein abundance, let's say even in a simple biofluid. I should not call it simple, but a biofluid like blood, because that's where most of our biomarker studies begin. So the most least abundant protein versus the most abundant protein, there's around 10 or 10 to 12 orders of magnitude difference in the amount. So any analytical technique has to deal with this kind of a challenge. Secondly, there is no equivalent of PCR for proteins. So whatever low amount is there in the cell or our tissue, you have to analyze using the technology that you have at hand. You cannot amplify just like PCR. Third, you have alternate splice forms of a gene, which makes it which can make different kinds of proteins using different combinations of introns and exons. And again, this is a huge challenge for proteomics because you have to identify each of these variants in the form of a protein. MRA alone is not sufficient. And then to add to it, there are hundreds and hundreds of post-translational modifications which cannot be directly deduced from a gene or an mRNA sequence directly. So you have to look at the protein species that are being formed and modified in real time. So with this, I come to the most introductory part of my talk, which is about what is the basics of mass spectrometry. This is the only technique that allows large-scale study of proteins without a bias wherein you will select the protein of interest and then start studying it. This is the rejoinder for systems biology in a proteomics context where you can study the whole proteome snapshot of a living organism in one go. So we sometimes fondly call it as the chemical balance for biology. So what does it mean? It means that mass spectrometry in the very basic sense is a chemical balance wherein you put some atoms or molecules and it tries to measure it. So before the soft ionization techniques, people were still trying to sequence proteins using mass spectrometry, but the problem was whenever peptides or proteins used to enter the mass spec, they used to break down. So the development of soft ionization methods, so soft means that they allowed the proteins or peptides to get ionized without breaking them inside the mass spectrometer. So MALDI and ESI were two of the techniques. MALDI stands for matrix associated laser desorption ionization. So in this technique, you have laser light hit upon a peptide mixture which is co-crystallized on a matrix. So this matrix is composed of a very low molecular weight volatile compound, something like cyanohydroxycinnamic acid. There are several other matrix compounds. So with this, you try to co-crystallize the peptides inside it, and when you hit the laser, the volatile compound evaporates, and it imparts a charge to the peptides. The peptide will take up the charge from this matrix and try to flow inside the mass spectrometer. And mass spectrometer is a chemical balance that cannot see anything except a charged particle. So if there is a charge to a particle, you can see it in the mass spectrometer. Otherwise, the molecule or atom is invisible to a mass spectrometer. Similarly, electrospray ionization has a gold tip needle, and it is coupled generally to a liquid chromatography column, wherein the analytes are being passed on to this by separation inside a liquid phase. And at the gold tip needle, there is high pressure and temperature, due to which the analyte keeps accumulating and a droplet forms. After, due to the high pressure and temperature, this droplet after sometimes will burst into fumes, and from the liquid analyte, it will also take up charge, and then it will be fed into the mass spectrometer. So both of these techniques actually revolutionized the field of biochemical mass spectrometry using proteins, and both were awarded Nobel Prize for their discoveries. So in general, this is a cartoonic representation of what a mass spectrometer is actually composed of. So you have on the first hand an ionizer, which could be ESI or MALDI. So this is to impart a charge to the peptides. In the middle, you have a mass analyzer, which could be based on different kind of geometry, which I'll talk in a very short way. But just to understand this, mass analyzer is something that tries to separate these different ions that are being injected into the mass spectrometer based on their mass to charge. So let's say if it is a time of flight, the smallest mass by charge will travel faster, and the heavier one will travel slower. The last part is a detector, which you can think of something like a galvanometer, wherein whatever charges impinge upon it, it will try to detect and give some amounts based on how much ions are being detected by this. And with this, you form the data, which is seen in the form of a mass spectrum. So there are different, as I said, different geometries of mass spectrometer analyzers will have different kind of formulas to calculate the mass by charge. The charge will be denoted as Q here because this is directly connected to physics. That's how this field developed. Although in biological mass spectrometry, we generally use the notation M by Z instead of M by Q. So different kind of mass analyzers will have different formulas to discern the mass to charge. And this is all inbuilt in the instrument, so you do not have to really calculate it by yourself. So these are the kind of peaks that you see after a mass spectrometer generates a mass spectrum. And this is MS1 spectrum. So one round of mass spectrometer will create peaks for one peptide. And I'll come to the details later. I'm just trying to focus on the working principles of mass spectrometer. So in mass spectrometer, specifically for biomolecules like proteins and peptides, we generally tend to use the monoisotopic mass. As the name suggests, this is the mass of a single isotope of a molecule. So, which means for biological molecules, this is the lowest mass in general because you may have C12 and some amount of C13, C14. As the less abundant isotopes are mostly radioactive and therefore their amounts are usually less than 5% or even 1% in nature. But you will, for the same molecule, you might see different peaks because one may have one extra deuterium or a C13 or a C14. Therefore, for sequencing, generally we only pick the monoisotopic mass. And there is this formula, the machine will tell you what is the M by Z. And if you know the charge state, which again machine tells you, you can multiply it by charge and subtract those many protons, because that is the amount they have taken up from the analyte, and then you can know what is the actual mass of, or the neutral mass of the peptide. There are different combination of instruments with different kind of ionization sources, although this is a bit older slide, and there are now even newer combinations where QTOF could be coupled to MALDI, but those are not that common. So now coming to actual proteomics, how do we use mass spectrometry for proteomics? So this includes two kind of techniques actually. One is called peptide mass fingerprinting, and the other is called tandem mass spectrometry or shotgun proteomics, or sometimes also called as bottom-up proteomics. So this is actually sequencing these days, but in earlier days, let's say 10, 15, 20 years ago, what was the major contributor to peptide sequencing was called as peptide mass fingerprinting. The technique is almost obsolete now, but in a way, it is also the first step in tandem mass spectrometry. So it's good to know as conceptual framework to understand how sequencing is being done these days. So that's how the whole process of proteomics begins. You have a sample which contains one or more proteins. For the simplicity of demonstration, I am only showing one. And you have a site-specific enzyme like trypsin, which specifically cleaves after lysine or arginine, and if not immediately followed by proline. So when you use this specific scissor called trypsin, you will generate different kind of fragments. And why we use trypsin? Because in general, for any genome, on an average, 12 to 15 is an average amino acid length after which a lysine or an arginine can occur. So we generate amenable size peptides for mass spectrometer to detect. So this is called peptide mass fingerprinting. So let's say there is some chemical noise also shown in black, although this would not be that much in height. It is just a demonstration purpose and not directly to scale. And let us assume all the yellow peaks, the broad yellow peaks shown here are the real peaks. So during mass spec sequencing, you will get some peaks, and we know that these peaks are coming from these peptides that we have just fragmented earlier. And this kind of forms a fingerprint for identifying the protein. And it was also mentioned earlier. So a single peak is just a mass. It may not be a unique signature to identify a protein, but several of these unique masses or even non-unique masses, when you try to find out, that forms a fingerprint which will clearly identify the protein. As an example, if I look at just my name, if I look for Amit, even in my own institute, I can find 5 people by the same. Almost 50 odd people will have this term in their name. But if I look at Yadav, again, I will find 2 or 3 people who will have that surname. But if I look all 3 in combination, it's a unique signature that will point to me. Similarly, protein masses are also like that. So even though protein masses are a fundamental property, they are not a unique one. That's why you need more than 3 or probably more masses to actually uniquely identify a signature, which will lead to a definitive protein identification. So in the whole process, how we see that a target hit is winning or a decoy hit is winning, the hit being the peptide hit. But in a separate search, since you're searching twice, you'll have one good hit from target, one good hit from decoy. And then you have to match whether which one is correct. So the formula, therefore, are slightly different. Again, this is a different representation of the same thing, and you see that in a concatenated data, in a separate database, you have for every spectrum S1 to Sn, you have a target score as well as a decoy score. But in a concatenated one, either a target score wins or a decoy score wins, and that based on that, the formula will be different, and you'll have different kind of distributions that can be represented. The basic fundamentals still remain the same, because if you put a threshold where you see only target hits, you will miss out some good hits because of the high scoring decoys. If you put the threshold somewhere near here where the least target hit is there, you will also include a lot of false positives. So you have to somehow choose between a good threshold that allows a lot of targets to be identified without much addition of false hits. You cannot for sure say which one of those are wrong, but when you say 1% FDR, only 1% of these may be wrong. So assuming that now you have put a very good statistical threshold, the idea was that you wanted to identify which proteins and not which peptides. The challenge here is this problem is similar to having a lot of jigsaw puzzles, all of which have some missing pieces because you do not identify a protein 100% because of the stochastic sampling during mass spec. Some peptide may not be digested, some may not be ionized well, some may not enter the mass spec at a proper time, may not be sequenced. So there are a lot of factors because of which you do not get the full picture, but you, if you get a unique identifying peptide, you can trace it to a protein. Now imagine you have hundreds and hundreds of jigsaw puzzles, each with missing pieces, and all put together, will you be able to synthesize things back? It's a very complicated problem, and this is just a cartoonic example where on the left hand side you see some peptides, and on the right hand side you see some proteins. You can see that protein A has 1 and 2 peptide which are both unique. They are not hitting to some other protein but only to A. So we know that protein A is there for sure. For protein B and C, you can see protein B has a unique peptide 3, but a peptide 4 is not unique. It can come from B as well as from C. Peptide 5 is unique, which can give C. So we are sure that B and C both should be present because both have a unique peptide, but peptide 4 alone would not have been able to tell that. If you look at protein D has a shared peptide 6 because it is also hitting to E. So protein D, we are not very sure whether it is present in the sample or not because the information is not enough to deduce this. So this is a lock and key kind of a problem wherein you can have multiple locks open some protein, multiple locks which can be opened by a single key. So now you have to reduce the minimum number of keys that can open all the locks. This is called a set covering problem in mathematics, and I'm just showing you some real data set. So this is human plasma where we have identified 100 odd proteins, and so these circles represent peptide and protein. So let us just look at this one. You have 4 proteins here, and these are the peptides that we have identified, and so none of them is a unique peptide. So you do not know for sure which protein is there. So in this method, what we do is we combine them, collapse this information into one protein group and choose one as the leader, which can explain all the peptides. Similarly, in the other one, you have 10 or 11 proteins, but only 4 peptides. Interestingly, these 4 peptides can equally come from these 5-6 proteins. So we again have to collapse them together and call it one a protein group. So when somebody says I have identified 100 proteins in a proteomics experiment, it actually means 100 protein groups. So this is something we developed which is called as a semantic protein grouping algorithm, which means meaningfully trying to collapse these peptides into protein groups, which we called as protein assembler. And now just to give you a taste of that whenever we are looking at diseases or evolution of disease, we cannot just depend on identification alone. We need to know what is the amount with which they are present in. As Lord Kelvin once remarked, to measure is to know. If you know something, then you must be able to measure it. That's what he said about the temperature scale, and that's how he developed the scale to measure temperature. And with this thought, as I also mentioned, that absence of evidence is not evidence of absence in proteomics. You may not see a peptide and therefore a protein, but you cannot definitively say that the protein was absent. It could be because of the stochastic sampling that you have lost that protein, but what you measure is more definitive. So that's the message here. So there's a huge quantitation bias in a proteomics experiment. So you could have this whole distribution under what is present in a sample. But because of our limitations in chromatography or separation sciences, we may have not all of them profiled in an experiment. Then again, because of our tools limitations, and let's say again, because of the data we generate, whether the data is good or not, you cannot identify everything, which is shown in the red part. So the blue part is what we miss out in identifications. But again, since whatever you identify may not have good enough quantity to be measured, it could be below limit or some other factors which could create an issue, we would be able to identify some proteins. So we can see there's a huge quantitation bias as we move from the whole proteome to what what is the fraction of proteome that we can quantify reliably. There are different approaches for quantification. It could be labeling based or label free based. And again, I'm only trying to give you a conceptual overview. Even if you do not understand them in detail, it's totally fine. You must just remember that there are techniques that can be used for quantification. So it could be either metabolic labeling like SALAC, or enzymatic labeling, or chemical labeling. Of course, chemical labeling is more popular because you can apply them for clinical samples, but sometimes limited by the multiplexing they can offer or how many samples you can process through them. Then label-free approaches could be either intensity-based, wherein you can look at the peptide intensities at MS1, or MS2-based spectral counting. So there's a potential bias at various steps in a proteomics experiment, and therefore, depending on the resources and the goal of the experiment, you must choose a strategy that is the best fit for purpose. So if you look at metabolic labeling, it may be costly, but since you're combining the healthy and disease state very early in the experiment, your potential for variation coming due to techniques is very low. But when you talk about chemical labeling, it's good when you're doing something like ICAT, but when you digest proteins to peptides and then you mix, which is most of the chemical labeling techniques today, again there is this inherent variation step wherein you are processing the proteins differently. Although we try to, all the researchers try to have all the parameters exactly same in the two experiments, but two runs of the same protocol, generally we are aware that do not give you the same results. So the peptide is the step where they are being mixed. Similarly, spiked standards are used for absolute quantitation as well. And again, depending on whether you inject it at protein stage or peptide stage, your variability may differ. And label free is the best in terms of economy, but not always the best in terms of the accuracy of quantitation. So generally, metabolic labeling is preferred wherever it can be used, and this is generally possible for amino acid labeling in cell cultures. Label free has a wide dynamic range, almost infinite theoretically, but the problem is that your run-to-run variability may sometimes give you false results. So should we use label free then? The answer is yes and no. It is free depending on if your labeling techniques are cost constrained, they can create a cost constraints, but methods will mostly be fit for purpose, and not any method will be all purpose all the time. So there are a lot of techniques in mass spectrometry, of which I will not be able to cover all. So you should consult an expert before planning. And again, I'm just mentioning some names here, the techniques that the algorithms that we use for label-free quantification. Just understand that either we use the area under the curve between two samples to identify what is the quantitation, or we do the counting of how many spectra we have sequenced for each protein, and that is called as spectral counting. When we use labeled approach, we generally tag with heavy isotopes and try to measure. So there is either an MS1 quantitation technique called SILAC, where we have a light, medium, and heavy isotope. So we can compare up to 3 biological states in one experiment. So these are 3 peptides from a same protein, and we can see them as triplet pairs in MS1. Or you could use MS quantitation, where you will have 4 to 6 to 8 kind of samples mixed together, and in MS1 you will only see one peak, you will not be able to differentiate, but when MS sequencing is done, you get the sequence ions, as well as in the lower mass region you will see what is the amount contributed by each of the biological samples, which is used for quantitation. These are called as reporter ions. There is also a hyperplexing or higher order multiplexing techniques, where sometimes people mix metabolic labeling as well as chemical labeling. So now each of the light, medium, and heavy tags will have 6 different tags, and now you can do 18 plex in one shot. Although this is very complicated, although we have tried this and also got a patent on this, but this is very difficult for a bioinformatic data analysis unless you know the principles of mass spectrometry. So in general, you get the raw data from mass spec. There are 2 forms of raw data. One is centroided, where you where the machine centroids all the monoisotopic envelopes into one, and you see exactly the peaks which will be peptides. And then you use for protein identification purpose mostly, and you have a profile data which is continuous data, and you, algorithms will try to filter out the reporter ions to do the quantitation at spectrum level. The identification and quantitation data now has to be merged together to see whether the same scan is identified as well as quantified, and based on that you pull out the peptides, and for each protein you look at how many peptides are there, try to merge these results, combine them into a protein quantitation report, which is later sent for statistical analysis. Okay, this I have already mentioned. Like you may have a light, medium, or heavy amino acid labeling. So you could use lysine and arginine generally for this kind of an experiment, wherein you use light, which is 0000 means normal lysine and arginine. Medium means you have their mass increased by a small amount, usually 6 daltons. And in heavy, you have lysine 8 and arginine 10, and you mix them in 1:1:1 ratio. So you can process 3 different states in one shot. You could also use a two-plex design and then try to have multiple SILAC states where you have one control but multiple medium states. So you have to now run each of these duplet pairs separately in different runs of the mass spec, and you can again try to increase the multiplexing, although it leads to more number of runs in the mass spec. So there are several algorithms for quantifying SILAC area calculation. Although in general you can directly use light, medium, and heavy across a protein, there is an algorithm called MaxQuant which will look at the 3D peak areas using mass by charge as well as intensity. And using this, you are able to quantify the SILAC ratios in a much better fashion. Again, there are several tools for quantifying SILAC as well. Then about the chemical labeling techniques, you can have 4 to 8 different isobaric tags. So you have a peptide, then you have a common mass of balancer plus reporter which will be added to each of these biological states. Since this total mass of reporter and balancer is always constant, you can tag up to 4 or 8. The principle remains roughly the same. So when you mix them together, the peptides after tagging, you will only see one peak in the mass spec. But when you try to fragment it in MSMS, you will see the contributing amount from these reporter tags, because the balancer is lost as a neutral loss. And we know that mass spec can only look at charged particles. So the balancer is not seen, but the reporter is seen. But the reporter will always carry a mass difference of 1 Dalton between these different biological states. So this is 8-plex iTrack. Earlier I was showing only 4-plex. This is another representation of the thing. We have a peptide and 1 Dalton mass difference of 4-plex iTrack, and again 1 Dalton mass difference of 8-plex iTrack. The total mass remains same, so the peptide is only seen as one peak in the mass spec. So this is again a similar representation that you mix, then you separate. When you send for mass spec, you see only one peak. When you break down in MSMS, you can see the amount contributed by each sample. So how is this area calculation done? So there are different ways, whether you can use some intensity based method or trapezoid method or baseline corrected method, where you also remove this baseline chemical noise from this area so that you get more accurate quantitation. And we have also developed certain tools for this, which is QuantWiz IQ. IQ stands for isobaric quantitator. So it can do for iTrack as well as TMT. TMT is another tandem mass tag, is another label like iTrack. Now there is a new technique coming up which is called data independent acquisition, wherein all the peptides are fragmented together. So this is a new concept which has come up in the last 6 to 8 years, and now more and more people are using it, wherein all the peptides are fragmented together, which may or may not be from the same protein, and swaths of 32 Dalton or 25 Daltons are fragmented at once. Now this creates a very highly complicated spectra. If you can see this spectra, you can see there are multiple colors here, and each color is a peptide fragment ion coming from a different kind of protein. So analyzing this is more complicated, but depending on the use, the challenge is that in swath, bioinformatic analysis is the most complicated part, and there are a lot of tools being developed for this purpose, but it is very good for dynamic range accuracy and coverage of the proteome and for biomarker identification. So people are really taking this up very fast. So thank you everyone, and thank you Dr. Rama for inviting me here. And although I have not talked about my work, the DBT and THIST has funded most of the work that I have done, although I was only trying to keep it to an educational purpose seminar. Thank you so much.
Thank you very much, Dr. Amit Kumar Yadav, for your excellent presentation. I'm sure many questions for you, but due to shortage of time, I think we may have to cut down just a few. So we have one question from a student from NJSM Pharmacy College, from Rishi Chaudhary. He wants to know the possible impact of advancement of in silico studies on animal studies in future.
I'm sorry, come again. Animal studies.
Animal studies, yeah.
So there are a lot of studies going on at the moment, because we know that animal studies do not always translate into clinical practice right away, because we need to do human studies as well. So and we also know the complications involved in a human study. So what advancement can bioinformatics provide is quite complex but straightforward in our center, because what we do is we have different animal models for different disease models, right? And we also know some data from clinical practice that, okay, for example, let's talk about cancer. We have cancer stage 1, 2, 3, and 4. And for a similar kind of a model developed, animal model developed for the studying that cancer, or let's say a metabolic disease, we'll segregate them into these stages. And depending on the molecular profiles that we can analyze using mass spectrometry, or let's say NGS, the technique does not always matter, but whatever chemical profiles we get from that animal model, we know that although it will not be directly translatable, but if we can calculate or normalize the age of this animal with the stage of the human, and calculate the molecular profile and try to match it with any clinical samples that we may have, we can accelerate the path to discovery. Because when we try to create a drug, and we try to give this drug to this animal model, and we see the effect, we can look at the molecular profile and try to predict whether the animal is getting better or not. Because only looking at a phenotype may not be relevant anymore. You need to assess a lot of biochemical parameters, and for such kind of studies, omics studies are a great way to assess these very quickly using a mass spectrometer or an NGS platform, where you know what is the genes that are expressed. You can find biomarkers quite quickly without working on human directly. And then the path to from animal model to human studies is quite shortened, and you have more data to actually say what you have postulated.
Okay, thank you very much. We have one more question. It's not exactly on mass spectrometry, but it's AI-based prediction. What is the future of AI-based protein fold prediction?
So, if you are aware, last week, I think it may not be more than 3-4 days, I guess, Google has come up with this AI-based prediction tool, AlphaFold, where they are saying that it is going to change everything. So, in my belief, for protein folding as well as for other kind of omics data analysis, AI is going to play a big role. And for protein folding, it is considered to be solved. I think we will wait and watch what is the kind of impact it has for drug discovery, and I also believe it will have a big impact. And but let us first wait how actually it is going to be used in clinic, because many times we have faltered because we sometimes read too much into a technological development, but it may not be ready for market as of now. So provided the predictions are being used directly in clinic, we still have a good promise, and they will play a very significant role. But let us wait and see how many of these are actually translated. Yeah. So I'll have a cautious tone there, but I'm pretty excited that these will play a very big role in all the fields. The only I think aspect to watch out would be that not all computer science people will be able to directly apply AI to all random biological problems. You need two kind of expertise. You need technical expertise in AI and ML, and you also need domain expertise in the field so that you know whatever parameter is being fed is actually biologically relevant, because these two things together can only make a huge clinical impact.
Yes. So we see a great future ahead. Thank you very much, Dr. Amit Kumar Yadav.
Thank you so
Thank you
much.
for an excellent presentation.
Thank you.
Our next speaker for the day is Dr. Shyam Peregu. Dr. Shyam Peregu is a PhD from University of Hyderabad. He did his PhD in 2014. His MTech in Bioinformatics and an MSc in Biochemistry from the University of Hyderabad, Telangana. His major focus of his thesis was on natural products and their role in treating breast cancer. He has utilized both in silico as well as in vivo approaches for his study. He has worked in various places, including the Jawaharlal Nehru Technology University, as well as National Institute of Nutrition, Hyderabad, as a scientist, before joining the Department of Biotechnology, NIT, Warangal. He has developed a diagnostic suite, a molecular property diagnostic suite, and developed several tools which are sponsored by the Government of India. So we welcome Dr. Shyam for this presentation.
Thank you, madam. Thank you for the wonderful introduction. So first of all, I would like to thank the Vice Chancellor of the NITTE and as well as the Director for the Research and Development, and inviting for me the Ramai Adiga. So today I am going to present my screen is visible to you?
Sir, I think you can give full screen.
Yeah, I gave, madam.
Yeah. It is visible. Yeah, please go ahead.
Yeah.
It is full screen.
Yeah. Today I am going to discuss about the current trends in the computational biology. So the bioinformatics and computational biology plays a important role in the current days, as already pointed out by the Vice Chancellor as well as the Indrani madam. So this is without bioinformatics applications, the current scenario, the most of the research articles are not accepting. So we need to add something related to the bioinformatics, systems biology, or computational biology. So how the bioinformatics will influence the hypothesis driven research, because earlier, so normally we used to do basis on the hypothesis, so we can conduct the laboratory experiments and we can get the results, or if our hypothesis is wrong, so we may waste the lakhs of the rupees and different types of the chemicals and our valuable time. So what the bioinformatics today is doing, so the bioinformatics initially itself, it assumes, it will predict whether the particular experiment will execute, whether the particular, the hypothesis, whatever we design, whatever the experimental design we have framed, that experiment will be executed properly or not, will be performed on computationally by using the various computational programs and computational algorithms, right? So before going into the lecture, so we will see the basic differences between the computational biology and bioinformatics. So what is the computational biology? The computational biology is the application of computational tools. These computational tools are developed by the programs, whether it is a C, C++, Java, Perl, Python, PHP, or whatever the programming language you are fluent. So you can develop your own program or algorithm or database, that computational tool and techniques which is used to use for the molecular biology problems. When I say the molecular biology is a vast. We have a number of applications we have. We have a genetics, we have a molecular biology, we have a microbiology, we have a biotechnology, we have a biochemistry, we have a forensic science, etc. This will be goes on, but everywhere the biotechnology contribution is there, right? So it enables the new ways of the study in the life sciences or basic sciences and allow us to analytic and predictive methodologies that will support enhances the laboratory work. When I say the bioinformatics and computational biology, they both are the synonyms as well as these both fields are multidisciplinary areas. So this is a combination of a computer science, biology, as well as the mathematics and statistics. In the another context, we also use physics and chemistry. When I talk about the drug discovery and design, then I need to work with the chemistry because the molecule synthesis can be done in the chemistry laboratories. When I wanted to do the molecular simulations and dynamics, I need to approach the classical mechanics as well as the Newtonian mechanics, right? So the biology is a integration with the computer science, statistics, mathematics, as well as the physics and chemistry, right? So computational biology is also termed bioinformatics, even though many practitioners that defines the bioinformatics somewhat narrower by restricting to the application of specialized software for deducing the meaningful biological information. That is about the computational biology. When I say the bioinformatics is explosive growth in the amount of the biological information, because when we study or when our professors are studying, all the experiments that are executed in the laboratories. So they used to write so many records and so many registers, but that data years together for so many files in a one research laboratory. So that is not computerized. Whenever you wanted to share your innovations to the global community, so we need to convert the data into an electronic form. So that is the way we can convert in the form of a publication, in the form of a research article, in the form of a manuscript. So but that data need to store in our databases. That databases we can call it as a biological databases. So what it will store the biological databases? Only biological information about the organism, about the gene, about the protein, about the function, about the structure. But we will develop. So you don't require the software developer. So when you learn any one of the programming language, then you can develop your own database, your own algorithm, your own software. But in reality, that software development is not that much easy because it is an integration with the 2 or 3 programmers is required, right? So then, but whenever we talk about the human genome, so we have 3,000 or 30,000 genes or 3 billion base pairs, right? So that human genome is completed. So whenever you wanted to do the human genome sequence analysis, the human genome sequence only took 10 years of the time. Now you understand how much time it will take to perform only one organism's genome to be sequenced. But there they have used the traditional sequencing methods like gene gun, bombardment gun, Sanger-Slade-Axey method, Maxwell-Gilbert methods, then followed by the automated method. So later, some of the technological developments they have started using, and then only they have started. So far, we have identified only 50% genes function. Remaining 50% genes function yet to identify from our human genome. So the improved technologies are developed that are called automated sequences, that are called next generation sequences. Okay. So whenever you do the organism's whole genome analysis or particular gene sequencing, when you perform by using the different type of the technology, then where you will store. So we have a platform called GenBank. So there we need to store whatever you found, that you can store it in the GenBank, that are also available in a NCBI database. But every 2 years, whatever the database size is there today, when you look at 2022, the size is doubling because so much of the work is submitting to the databases, because everybody is aware, everybody using the modern technological applications to be sequenced, to be identified of a organism's function, character and structures, right? Some of the different type of the definitions for the bioinformatics as well as the computational biology, because we look at the bioinformaticians define their own field in a very different terms, because this is simply I wanted to say the bioinformatics is the use of the information technology or computer science to solve the biological problems, to store the data, to develop the algorithm, to develop the software, to be analyzed by molecular biology or biological problems, right? So that is the simplest step. But when you wanted to write in your examination pointer, we need a particular statement. So bioinformatics is an integration of a mathematical, statistical, computer science. Some of them may say the information technology. So these are to analyze the biological, biochemical or biophysical data, right? That is the data we need to analyze. So the bioinformatics is the study of the biological information as is processed from its storage site in the genome to the various gene products in the cell. It involves the creating and development of advanced information and computational technologies for problems in molecular biology. So the bioinformatics, in another aspect, the bioinformatics is specifically refers to search and use the patterns and structure in the biological data and the development of the new methods for the database access. Okay. So another few more definitions. So some people make the difference between the bioinformatics and computational biology. So that are bioinformatics is specifically refers to the search and use of the patterns, motifs, domains, and structure in the biological data and the development of new methods for the database access, database control, database development. When I say the computational biology is more frequently used to refer the physical and mathematical simulations of biological processes. So bioinformatics will be used for a research, development, as well as the application of the computational tools and approaches for the expanding the use of the biological, medical, biomedical, behavioral, as well as health data, including those to acquire, store, analyze, archive, and organize, and whatever the data you have, we need to visualize the data. That will take care about bioinformatics. So in the computational biology, the development and application of a data analytical tools and theoretical methods that are mathematical modeling and computational simulation techniques to the study of the biological information and behavioral systems. How could I define it? The development and applications of methods from the computer science, statistics, mathematics, and physics and chemistry to analyze biomolecular or molecular biology data. So this is about the computational biology as well as the bioinformatics. So this is a traditional slide because wherever I go, so I carry this particular slide. So as a human being, our body contains different organs, different tissues, and different mechanisms of action, signaling mechanisms, and various metabolisms. Whatever it may be, just you may take tissue or blood sample, then you extract or isolate the cell. That cell contains mitochondria, nucleus, Golgi apparatus, cell membrane or plasma membrane, cilia, endoplasmic reticulum, smooth and rough. So when I look at the nucleus, what is there in the nucleus? Nucleus containing the chromosome. The chromosome consists of telomeres, centromeres, chiasma, etc., etc. So if I wanted to talk other aspects, alleles, locus will be there, right? So from that chromosome, the histoproteins will bind to DNA. That DNA has the double helical structure. These DNA containing adenine, guanine, cytosine, thymine. So purines and pyrimidines, that are the nucleotides. If nitrogen base, sugar base, and ribose sugar, nitrogen, and phosphate, these three components will be present in the nucleotides. So we have a genome. In that genome, we will extract the gene. The gene will be having DNA. The DNA is a heritable characters for ours to our offsprings, our parents to ours. The heritable characters will be carried forward from the one generation to the another generation, or sometimes it may be skipped. So this DNA will be transcribed into a RNA, and this RNA will be translated into a protein. So this is about the central dogma of molecular biology: DNA to RNA, RNA to protein. So as all we know, so but our intention, when I say the bioinformatician and computational biologist, I will locate from the genome to the protein. By side, which metabolism, which pathway is involved by particular gene, particular DNA, particular protein. What is the mechanism of action that are doing? So when you wanted to do the DNA analysis, we have two type of methods. One is the qualitative analysis, another one is the quantitative analysis. When I say the quality, our product should be quality. How many base pairs that has, what is the size of a particular gene. So that can be identified by the various methods are there. The simplest method is the gel filtration. So when I say the quantitative, just only we require the purity of the product, how much quantity is there. So basis on the optical density, that optical density will be measured by using the calorimeter as spectrophotometer. So this is the DNA. Whenever you have a DNA will be extracted, and that extracted DNA will be used by the various methods. That methods are, the DNA can be estimated by the perchloric acid method, that is a PCA method. Another one is called diphenylamine method. So ethidium bromide method and UV spectrophotometric method, and the last one is fluorescence method. So these are the 5 methods which we use for the estimation of a DNA. These are the traditional methods, but there are the advanced methods are there. So we'll go discuss in the next slides. So that is about the DNA. Then the RNA. So when I wanted to estimate the total RNA from the given sample, like either blood or tissue, so we approach the perchloric acid method or arsenal assay or spectrophotometric assay. So when I wanted to estimate the protein, so spectrophotometric analysis can be done. If I wanted to analyze the total protein estimation or total nitrogen estimation by using the Geldal method or Geldal digestion or distillation and titration methods will be used. It depends upon the macromolecules and micromolecules. So calorimetric analysis can be performed for the identification of the estimation of the proteins. So in the calorimetric analysis, if you are undergraduate and postgraduate, you might have used the biuret method, Lowry method, or Bignonic acid method, or Barfoot method. So these are the 4 methods which we use for the calorimetric analysis. The another one is the solid phase protein assay will be used, and gas combustion method will be used, and the HPLC, different type of the chromatic methods also will be used. So that is about the traditional method. So somewhat we go the forward direction because, as I said, the trends, current trends. So what are the latest methods which we are using? So somehow advanced methods are the chemical cleavage methods that are called maximum Gilbert method. These that in the memory of the 2 scientists, that is Alan Maxim and Walter Gilbert, has developed the chemical cleavage method for the sequencing of a particular DNA. So another method is called enzymatic chain termination method, that is developed by the Sangers. That is in the memory of the Sangers dideoxy method will be named, and that will be used as a enzymatic method, and Edmund degradation also a method. And these are the 2 basic methods, and somewhat advanced method other than the chemical as well as the enzymatic method, the automated sequences came. And somewhat advanced method other than to the automated sequencing methods are high throughput sequences. So these high throughput sequences will be used the various type of the technologies, including the next generation sequencing. So normally we have a DNA, RNA, protein. This is the oldest method. So the classical methods or classical sequencing methods are the Sanger's dideoxy method, that are the automation methods, and base calling, and fragmentation of a sequence, and quality scoring, shotgun sequencing, and gene sequencing method, gene bombardment gun method, and assembly and finishing methods are the classical sequencing method. Later, the next generation sequence is Java, that is called used to use for the virus sequencing, CRT, Absalate, and this will be used to use for the resequencing of the sequences and epigenetics and RNA sequencing analysis. In the third generation, this SMRT and nanopores came into the picture. The current scenario, we are advanced than the next generation sequencing. We are using the nanopore technologies. So these oldest methods are the context for the technological as well as the biological aspect. So the classical methods are used as a concept that are developed by the chemistry, enzymes, physics, as well as the computers. So this is the next generation method is a contemporary method, and third generation methods are contemplation method. That is a contemporary method, and this is the contemplation method, right? So this is a futuristic method which we are using in the present scenario and modern days, the nanopores and not. And but when you come from the context to contemplation, the cost will be dramatically increases. Okay. So this is okay. You are talking about the DNA sequence or gene sequencing. But what do you do when you do the sequencing? So whatever in my laboratory, I may do the sequence, I may do the experiment. During my experiment, I found some nucleotides or ATGCs in different permutations and the combinations, but I don't know what kind of the function it may do. So for that reason, I need to perform the similarity searches. So my sequence is similar to what kind of organism. Once it is organism, is it? What kind of the function, what kind of the structural similarity, what kind of purpose this sequence is used. Basis on the sequence, we will compare by using the bioinformatics tools and softwares. So we can perform the sequence analysis by using the pairwise and multiple sequence alignment as well as the hierarchical methods that will be identified the sequence similarity, identity, and homologous. When I say the homologous and similarity and others, so before that we have sequenced some of the model organisms. That model organisms are the chimpanzee. When you compare with the humans, it is a 98.33% are identical. The musculus mouse, 85% is identical. Zebrafish is 85% identical. Arabidopsis thaliana is a 26% identical. Yeast, Saccharomyces cerevisiae is 23% identical. Roundworm is a 21% identical, and bacteria, that is E. coli, Escherichia coli is 7%. So that is the reason we are similar, we are identical to the some of the known organisms, because whenever you wanted to perform the laboratory experiments, especially when you wanted to perform on animal studies, so we may not conduct experiments on all type of the organisms. So we can choose whatever the organism sequence is done and whatever the organism's reproductive age is fast, those organisms, and whatever the organism that is specifically or suitable to our experimental conditions, we can select. Basis on that, we can select the organisms. So this is the phylogenetic tree. When you look at the invertebrates and vertebrates and whatever the model organisms as I spelled out, so these are the evolutionary tree we have constructed, and during the evolution we are identical or similar, and we are changed our genetic information from the single branching point or bifurcation has started from the different points. Okay? So this is the similarity of humans, or sequence similarity with the different model organisms with humans. Okay. So this is an interesting way. So Jane Goodall has performed the various experiments. Actually, she is the environmentalist, and she performed various experiments on chimpanzees, and she used to speak to chimpanzees, and chimpanzees also speak to her, because she spent more than 30 to 40 years of her life with the chimpanzees. So here, I have shown the two sequences of the myoglobin, one from the human, that is from the Jane Goodall, and another sequence from the chimpanzee. So these two sequences, the human sequence is the first one, that is a red or pink one. The another one is called blue one or cyan color, or the chimpanzee sequences. So the total protein length which I have taken is 153 residues. But when you compare the two sequences, that are the homologous sequences, that homologous sequences have the same identity. A by A, B by B, C by C, D by D, E by E, F by F, G by G, H by H, I by I, K by K. That is called identity. When I say the similarity, R by H is identity, similarity. K by H is similarity. What is K? What is R, sir? So we'll discuss. Look at here the two sequences. Jane Goodall has seated, where chimpanzee is seated. How the former president of America as expressing as how the physical exposures and body different type of the postures, your chimpanzee also have the same posture. So if you want to compare one president, why not the another president? Right? So this is what the identical. So when I say orally, you may not believe this is what the physical. This is about the sequence which we computationally compare the sequences of the human with the another organisms. That may be any organism we will compare. Then we can trace out the percentage of the similarity, percentage of the identity. Clear? So when I say this, these all are the amino acids. Amino acids are classified according to their functions. Acidic amino acids, basic amino acids, aromatic amino acids, hydroxylic amino acid, hydrophobic amino acids, sulfur containing amino acids, charged amino acids. Right? Basis on that. Arginine, for example, see here, acidic amino acid, as per Jane, N. Then Q is a glutamate. Wherever the sequence containing the N, that may, in the second line, may come the Q. That is similarity. Because the same type of the function it will give. Because the two amino acids are related to a one functional group. Or NN is there identical, NQ is there similar. I am talking about only functional aspect, about the amino acids. Okay. For example, you have a histidine, lysine and arginine. H by R or K by R or K by H. These are the basic amino acids. Clear? Or I have a cysteine and methionine. These two amino acids are sulfur containing amino acids. right? So this is the way we do compare the sequences one with the others, and we can identify the sequence similarity and functions. So the bioinformatics and computational biology uses the molecular biology, genetics, biochemistry, biophysics, and ecology and evolution, and this will perform the genomics, proteomics, and metabolomics, image analysis, data management, numerical analysis, that is a regression and correlation, statistics, and mathematics, and different type of algorithms. These algorithms, these softwares, and these programs will assist us in analysis of your biological data, right? So the genomics here, the high throughput DNA sequencing, as I already told some of the sequencing methods, the first high throughput sequencing genomics technology was automated, that is the DNA sequencing was initiated in the 1990 and it was finished in the 2000. So almost 10 years of the time it has taken. So during this period, 1995, the Benton and Hamilton used the whole genome shotgun sequencing strategy to sequence the genome of mycoplasma as well as the hemophilia synthetic. Later, in the September 1999, Cellular Genomics, this is a software and computational tools, has completed the sequencing of the Drosophila melanogaster genome by using the shotgun method and different algorithms. Okay, so they were identified 3,000 million or billion base pair human genome sequence was generated in a completion between the publicly funded human genome. So what are the organizations that are contributed? Human National Institute of Health, National Laboratory of Medicine, as well as National Center for Biotechnology Information. So these are the organizations are involved in completion of a human genome, and they were identified with the human genome sequence. These are the automated sequencing methods, and they used to do the confocal detections between the mega-base sequences by using the fluorescently labeled DNA samples. And how about now? So dramatically, the various technologies are coming into the market. They are coming up with the new technologies and new programs. The technologies are fasten up the sequencing strategies. Okay, so whenever the new technology will comes, the cost also will be increases. Cost will be increases. Computational time will be reduced. Okay. This is the sample collection and experimental design, and it will be performed the various type of the experiments. So this is a contribution for the different factors to overall cost of the sequencing projects across time. The new era of a molecular biology as well as a biotechnology. We are using the microarrays for identification of a gene expression. We are using the two-dimensional cellular forces for the gene expression or the protein expression analysis. The multi-level maps, that is called genetic variants and genetic analysis, that are the physical and the sequence and annotation methods. The various type of the network strategies which we use, like tree method, graph theory, and topology for the identification of a protein-protein interaction and gene-protein interactions. So subsequently, we have a cross-species interrelationship. Some of the species is having the same similarities and same functionalities and same relationships. same pathways they may associate. That is the time we need to do the chromosome organization as well as the homologous genes identification. So the use of the bioinformatics tools or computational biology tools are the crucial. The new biotechnology tools generate the explosive growth in the amount of the biological data. This biological data is impossible to analyze the data by manually. If you wanted to analyze your own data, your own research data, you are taking to finish up your own doctorate, you are taking the 5 years of the time. Let us assume the global scenario. How much time it requires, how many years will take to complete the data. So that is the reason the bioinformatics will play a crucial role to analyze the data that is generated by the different laboratory researchers. By using the computational algorithms and softwares and programs will be reduce the laborious work as well as the computational. So novel mathematical method, novel statistical methods, novel algorithms, novel computational methods are available, and some of them are coming up with the new methods, and they will do the sequence analysis as well as the functional prediction and structural prediction. The day by day, the sequenced organisms like bacteria, eukaryota, archaea, and their organisms, their different species, are dramatically increasing the sequencing of a organism. This is a country-wise, the sequencing projects are going on. And this is also so far the completed genome projects so far from the 1997 to 2020. So some of them, the red color one, the drafts are ready, and some of them are already completed. Okay, so this is about the total genomes are identified so far during the year and their genome size with respect to. So this is about the complete human, chimpanzee, mouse, dog, and Platypus, rice and mosquito, Plasmodium falciparum, yeast, E. coli, and HIV. Their classification and their genome size and estimated number of protein coding genes. Okay, so this is about the comparison from the human to the HIV virus. So this is the protein data bank have yearly growth, and day by day there's a number of the protein structures, protein crystallized structures by using the NMR, by using the X-ray. If not possible by these two technologies, then we are using the bioinformatics tools that are the homology modeling, comparative modeling, ab initio modeling, threading will be used, and we can predict the structure and we can deposit into a PDB database. So these are the PDB structural database containing the so many PDB entries by using the X-ray, NMR, electron microscopy, and others. The total as on today 1,72,175 PDB structures that are available in PDB database. So how to handle the data when you look at, if you have a time, so you can go to the NCBI database and look at the database, how many genes, how many genomes data that is available in the database. So these are the 3 billion pieces when you talk about the human. Pieces of genes you have. How to analyze, how to handle the 3 billion pieces. Okay, so that is big. Then we are using, you have a big genome, then we are using the big data analytics, big data tools to solve our problems. So whenever you wanted to do by applying the algorithms to analyze the genomics data, so we just normally clone a gene, then we need to already, this gene is available in database. If it is available, what is the accession number, what is the annotation? So we need to check. Then, is my sequence is similar, whatever I have cloned, whether that sequence is similar in terms to the bacteria, in terms to the fungus, in terms to the organ, in terms to the sequence. What is the percentage of identity? What is the family? And is my sequences conserved to the particular sequence, particular organism, particular family? What is the alignment? What is the domain? And what is the evolutionary relationship between my sequence, my gene, to the other genes, to the other organisms' genes? So then we can construct a phylogenetic tree and we can identify the evolutionary relationship, right? So similarly, the protein characteristics will be identified, that is a sublocalization and solubility and three-dimensional folds, and other informations like expression profiles and mutation analysis can be performed. So when I talk about the databases, we have a biological databases. That biological databases are divided into a structural databases, sequence databases, bibliographic databases, and specialized databases. So in these databases, we are going to look at the sequence databases. In the sequence databases, we have two types. One is the DNA sequence databases, another one is called protein sequence databases. When you look at the DNA sequence databases, we have a GenBank, we have a DDBJ, we have TIGR, we have a yeast, we have a microbes, and we have some of them are called the ESTS databases. And moreover, we have a National Center for Biotechnology Information database for the analysis or searching of a sequence information related to the DNA. Similarly, you have a protein databases in terms of amino acid. We have a XPASI, that is the Expert Protein Analysis System. That is a SysProt database, or it is available in a UniProt. So here we have a PIR database, that is a protein identification resource. And we have a translated databases called Translated European Molecular Biology Laboratory Database. And we also use the NCBI database for the gene peptides, that is coding regions will be translated into your proteins. So when you talk about the structural databases related to the protein, that is a PDB. So that is a protein data bank. So whatever the databases you have, whatever the databases you are holding the data, that databases can hold the information in the form of a sequences. That sequences have the formats like ASL format, flat format, FASTA format, GenBank, EMBL, EBL, PDB, XML, or HDF5. Some of the other formats are also available. These formats will be used to use. These formats have the header content, biological function content, and structural content. Okay. So how do we clone? Which is a positional cloning. So we have a genetic aspects. In this genetic aspect, we have a pedigree tree. So we have a different type of the samples. We may add the genetic markers and where it is located on a particular chromosome, what type of the mutation, what type of the disease, what type of the mutation is available. That mutation can be, how can it be identified by using the different type of the vectors like YACS and BACS, and the transcript identification can be performed by using the different type of the technologies. If it is a mutant, how do you identify the mutant and mutation search? Then how do you get the sample and how do you do the analysis? So for this purpose, we have an advanced method called positional cloning. That positional cloning can be used by the next generation sequencing methods. So this next generation sequencing method, we have a evolutionary sequencing technologies we have. In this evolutionary sequencing technologies, we have a different type of the domains and different type of the genes. The Gs are, first G is the Sanger-Dieckx method, the first generation. Second generation, we have a massive parallel sequencing method that are the Illumina, Alexa, and Rache, and Applied Biosystems, and Ion Torrent. Because these are the commercial platforms, and that is what I am not talking about the much, because every company have their own pros and cons. And because I'm not a company person to talk about all the pros and cons about the different type of the products. And in third generation, the massive parallel sequencing with the single nucleotide polymorphisms and single molecule sequencing that are available with the specific biosciences that is called as a PacBio and Helicos, this Helicos biosystem are available for the genetic analysis. The fourth generation systems, which I already said, the current scenario is using the nanopore technology. That nanopore technology is available with the mini ion, grid ion, as well as the Oxford nanopore technologies. But the cost is as an average, the 2 crores to 4 crores, the investment cost. So these are the novel identification of genes for the different type of functions from the 1986 to 2020. So this is also a exome sequencing for the different type of the functionals, and we have a next generation sequencing strategy. This next generation sequencing strategy will be used for the whole genome sequencing as well as the exome sequencing as well as the target sequencing. So how do you do the target sequencing? The sequencing of a particular group of genes that are responsible for a phenotype. So we need to identify what are the genes that are involved. For example, the cardiomyopathy, deafness or blindness or diabetes or cancer or whatever the disease you wanted to target. So the technology will be used, and inherited diseases panel we need to identify whether any inheritable characters are associated or influenced by particular disease. That inherited panel will be examined. Then we can do the target analysis for a particular disease and particular gene and particular tissue, particular location. Then exome sequence can be used for the clinical utility of a biological sample. That will be screened for a mutational analysis with the unknown phenotypes. So another one, we need to do the research with the identification of new genes for the Mendelian phenotypes that is carried out by the genome-wide association studies, and we need to perform the cancer genetics research. The whole exome versus the whole genome analysis. So you have a large amount of the data, but 1% of the human exome need to be identified with the untranslated regions from the 5' and 3', and non-coding regions and coding regions, and introns and promoters, and exons regions, and splicing points, and stop regions, everything we need to identify by the genome analysis. So while doing that, we can know what kind of the experiments it will be performed. So normally we have a genome. The genome DNA sequence will be taken from the either blood sample or tissue. Then we will perform the extraction methods. Then we isolate the DNA, genome DNA sample, and we can fragment it, whether it is a 9 residue length or 11 residue length or 14 residues. So that is called sharpmers or K-mers. That is, a DNA will be fragmented, and we will be added the adapters. That is, adapter will be ligated to the whatever the DNA we have fragmented, and forward primer and reverse primers will be used, and the particular DNA will be amplified by using the PCR. That the way we can generate the fragments. That is a fragment library. Then we can capture the exome library. So then we can exome capture kit is available. Then we do the hybridization. From the hybridization, we can select and we can amplify and we can barcode the sequences and we can perform the next generation sequencing. So this next generation sequence will be used different type of the technologies, as I already said. And the NGS analysis is completely programming methods. So here we used to use the sequence is normally in the bioinformatics and computational biology we can say the FASTA sequence, but here the sequence will be fragmented into a short length, that is the 9 residue length, 13 residue length and 16 residue length. So that sequence will be called as a FASTA-Q format. And this FASTA-Q format will be used as a quality control sequences. Then this quality control sequences will be check the quality of a particular sequence, and it will be analyzed, identify the different pairs, the pair 1, pair 2, pair 3, pair 4 sequences. You can do the alignments, that is called sequencing methods, by using the pairwise alignment like that. So we used to use the BOT tools and BWA tools, and that they use the SAM tools. These SAM tools, whatever the sequence shortness you have, and that will be converted into a SAM to BAM and BAM to the paired sequences with the index. So whenever you have a sequences, whenever you have added the barcoding and other things, then it will be prepared as a variants. These variants will be pre-processed by, if any duplicates are available, by unknowingly, so that duplicates will be removed and it will be re-performed the sequencing and that realign the sequences. Any insertions, any deletions are required, any mutations are there, that will be deleted and appropriate mutation will be inserted, appropriate residues will be inserted without losing the functions. So that will be recalibrated by using the GAT-K tools, and once it is done, it will be analyzed by the SAM tools and NGS rich analysis by the variant callings, and that will be do the annotation as well as the parsing. So whenever we do the parsing and whenever you do annotation with your sequences with the other known sequences, because we have a database, that database containing the number of the organisms which sequence is known, which genes function is already known, that is available in the database, and whatever we newly perform, that will be used the database search, and basis on the database search we will retrieve the reports, and that reports will be used for the quality, alignment and capture and variants. Right? So this is the work process for the NGS analysis. For the pre-processing, we used to use the FASTA-Q, FASTA-C and FASTA-X in prime sequences. For the mapping purposes, we used to use the BOT and BWA and TopNet and GMAP. And different type of the assemblers which we have, the Velvet, Mira, Trinity, SOAP and other tools. And other SAM processing tools, SAM tools are available, and Picard and GAT-K tools are there available. And formats will be used for the conversion purposes, where the variant call conversions and FASTA-Q conversions and SAM to BAM conversions and bit conversions and GFF conversions will be used for the different type of the approaches in the NGS scenario. So these are the different type of the technologies. Whatever we do traditionally, we do the experimental design and we perform the genomic analysis. The genomics may be a metagenomics, and then from that we do the transcriptome analysis and we do the whole genome analysis. So sample type, whether it is a human sample, whether soil, whether food, whether water, or any single organism. Then nucleic acid extraction can be done by the manually or automated, and different type of the technologies will be used. As said, the iSequence and iSequence, ion torrent, or 454 pyrosequencing, Upsalid, or PacBio. So whenever you do the NGS analysis, the data analysis will be plays a crucial role. So that analysis can be done by the deionizing and sequence assembly, gene annotation and statistical analysis methods. So this is the way the different type of the methods like Illumina, Roche, Ion Torrent, and Solid. So different type of the library preparations they use, and template preparation and sequencing strategies. So one person will use the sequencing by synthesis of a fluorescence detection and pH detections and ligation method and synthesis of a pre-energy, right? So these are the by basically they used to use the template preparations by using the emulsion PCR, but Illumina will use the bridge amplification. Okay, so these are the technologies which we have, because whenever we wanted to talk about the only NGS, it required 3 to 4 hours time, because the various variant preparations, call preparations, and everything will be available. So that is the problem. So that is the reason. So I have discussed about the only current scenario in the computational biology as well as the bioinformatics. So by this, I will stop my presentation. If you have any queries, you please feel to ask me. So I may answer, if I know, I may answer your queries. Thank you.
Thank you very much, Dr. Shah. We just wait for a few seconds for any questions. Are there any questions? Okay. So I think everybody is quite hungry and there are no questions. Maybe we'll call you once again and then there'll be more questions.
Sure, ma'am.
Yeah, yeah. We'll wait for a few minutes. Yeah. Yeah. Dr. Shyam, I have one question.
Yes.
Can you tell which is the most, throw some light on the most popular platforms like for sequencing, which is currently in use?
But madam, the thing is that whether you wanted to use the NGS, normally we are using, because it's a financial matter, because whenever you wanted to purchase Illumina, you need to spend almost 1.2 crores. Absolute is 1.4 crores. When you talk about the PacBio, is almost 3.2 crores. Whenever you wanted to use Nanocore, it's 4 crores. So this is a financial matters are associated, but the quality and analysis is good when compared to the Illumina to the Nanocore. So last stage of the sample, last stage of the sequence will be very accurate in the Nanocore when you compare to the Absolute Illumina and 450 AB. So it depends upon the starters, or you may do the by what they call by paying the money and you perform the sequencing. Then you can use MI sequencing or Illumina sequencing.
Thank you very much. So for our participants, the feedback link will be given there. You have to fill up the feedback link and immediately the certificate will be generated. So thank you very much, Dr. Shah, and I'm sure we'll have you for talks in future, more such talks in future. Thank you very much.
Thank you, ma'am. Thank you for giving the opportunity, madam.
Yeah. So for the participants, please fill up the link. Has it? Yeah. So you can just fill the feedback forms and the certificate will be generated. So thank you everybody, and we look forward for more such future interesting talks. And thank you, Indrani ma'am, and the resource person special thank you to one and all.
Thank you, Raman.
Thank you.